Alright, well thanks to everybody for coming today to have a look at my talk on dementia. In a second I'm going to change the slide and I'm going to keep the next slide on for 30 seconds and there's going to be a sequence of numbers that I'm going to want you to remember. So try really hard to remember them. Okay, 3, 2, 1. Alright, time's up. So I suppose now you're wondering, how did you remember those numbers? So let me just tell you briefly. First, light entered your eye and you saw the numbers on the board. And then that light hit the retina and receptors in the back of the retina made sense of that light and turned them into electrical impulses. These impulses are then carried down through neurons across these synapses, and neurotransmitter is used to transfer the impulse from one neuron across a gap into the next neuron. And this happens many millions of times, all the way through the brain until you get to where your memory is stored. In this case we're talking short-term memory, and that short-term memory is stored in the prefrontal cortex over here. Another way of remembering how short-term memory is found, how you remember stuff using short-term memory, is if you imagine you're walking down a beach, for instance, and you leave footprints in your wake. And then whenever you want to find that memory again, or you want to retrieve it, you want to remember those numbers, you look back at your footprints and you can see that there's a trail of displaced sand on the ground. And that's how you can trace, it's called a memory trace, you can trace your memory from A to B. It's a unique sequence. The scientific way of how this happens is that specific amounts of neurotransmitter are depleted when you have an impulse crossing synapse. And there's also quite a unique sequence of neurons that are used to transfer what you see into your prefrontal cortex A to B. So what is dementia? Dementia, let's break it down. The word dement. The first bit, de, means without, and ment means mind in Latin. So dementia means without mind. This is the only time where I'm going to have loads and loads of text on the board. So dementia is the loss of global cognitive ability as a result of neurodegeneration, or the progressive loss of structure or function of neurons, including neuron death. Now, we can have a look here that there are quite a few types of dementia, and that's quite a big problem in the UK. Now, I know that I've got the table over here and it does say US, but this happens in the UK as well. At the moment, we've got all these different types of dementia. You've got Huntington's, Parkinson's, Alzheimer's, and in a population of 305 million, 4 million people have Alzheimer's. Now, obviously I said earlier this is the US, but it applies to the UK because in 50 years' time we're going to have quite a lot more people that are over the age of 65 in our population, and so these numbers are going to increase quite a lot, maybe even exponentially. What I'm going to focus on for the moment is Alzheimer's, because 4,000 people with Alzheimer's, it accounts for about 65% of all people that have dementia, all the cases. And that's a massive problem if you consider what the current cures are for dementia at the moment. The problems are that there aren't actually any cures for dementia. Right now you've got people in families who get dementia and then they're really old, and then the families get fed up and just bung them in a care home. That's not right, is it? You have therapeutic treatment, which is basically they learn to live with their dementia, they learn to live with the symptoms, the memory loss, the lack of movement. And in certain care homes, they can design the care home to make objects stand out. So, for instance, they'll design the floor of the care home to be so you can't get lost on it. Also, you have got some drugs which can slow the onset of some types of dementia, like Alzheimer's. But, for instance, in Parkinson's, that's 1.5 million people where you have no drugs that can slow down Parkinson's at all. So these cures don't exist. That's a big, big problem. Now onto the sciencey part of my presentation, a breakdown of Alzheimer's. In Alzheimer's, you have unusual deposits of two proteins, beta amyloid over here, and tau proteins. Usually in the regular person, they're soluble. So neurons, brains, whatever, they can produce these proteins, but then they're soluble, they're broken down, they're harmless. But in Alzheimer's, these clump together and they become quite insoluble. And then they form these extracellular deposits over here, or intracellular deposits of tau proteins, which have quite a few problems with each of them. In tauopathies, which is basically what happens to the tau proteins, inside the cell, you have the cell's transport system, which is called a microtubule inside the cell. And you can see this as maybe a train track. From A to B, you get nutrients sent from dendrites over here all the way down the axon, and that's how the bits over here get their nutrients. Now, the tau proteins in normal people hold the microtubule together, all these little blue bits hold the microtubule together. But in people with Alzheimer's, the tau proteins are misfolded, and if you do biology, you'll know that misfolded proteins don't behave in the same way as regular proteins work. And as a result, you've got this accumulation of misfolded tau across the whole microtubule. And the cell notices this. The cell's like, hey, you know, these are wrongly folded. These won't work properly. So it tries to get rid of those proteins by invoking something called the unfolded protein response. And essentially, you have these things called kinases, and they hyperphosphorylate. They add loads of phosphate groups to a tau protein. And in a nutshell, this means that the proteins disintegrate from the microtubule. Imagine this as the working microtubule as a thick woolly jumper. But in the disintegrating microtubule, all the fibres have come loose. You've like pulled a fibre apart and the jumper's just disintegrated. That's what happens with the tau proteins. The tau proteins dissociate themselves from the microtubules and clump together outside, forming these deposits over here. And as you can see now, like I said with jumpers, the microtubules just fall apart. They're not held together by these proteins anymore. So how are you supposed to get from A to B if the protein's gone? If the main train track, if the main transport system of your cell is gone? So this can lead in the neuron becoming diseased or even dying. The next thing is called proteopathy, and this is how beta amyloid deposits are formed. In a normal human, you have a protein called amyloid precursor, and this is a transmembrane integral protein, which is in the cell membrane over here. And as you can see, with a normal person, you have an enzyme called alpha secretase, which cuts the enzyme and creates two bits. No, it cuts the protein, the enzyme cuts the protein, and you get two bits of the protein. The first bit that's cut floats outside of the cell, and this has neuroprotective properties, and it also promotes neuroplasticity, which is how you relocate some memories if stuff goes wrong. But unfortunately, in Alzheimer's patients, you don't have alpha secretase, you have something called beta secretase and gamma secretase. And these two enzymes are different from the first one because they cut APP in two different places. Now, instead of having this APP stub alpha, which is the first part of the protein that goes out into the cell and helps everything, you have two different things. You have the amyloid precursor protein beta stub and beta amyloid fragments. This stub over here is completely different from the first one, and as a result doesn't have the properties of the first one. It doesn't have the properties of neuroprotection or neuroplasticity, and it reduces the above things. And beta amyloid, which is this thing here, the yellow thing, usually it's soluble, like I said before, but in people with Alzheimer's, they're cut, they assemble together into massive insoluble plaques, and they're neurotoxic. They could even kill the cell here, or they could kill other cells around it. It's not looking good for people with Alzheimer's, basically. Also in familial Alzheimer's, which is when people have a genetic condition, you can have a mutation in the gene that is also responsible for the actual synthesis of APP in the first place. This molecule could be twice as long. And if it's twice as long, you can have twice as many beta and gamma secretase enzymes cutting beta amyloid, which means that you have twice as many beta amyloid deposits, and Alzheimer's starts going off potentially twice as fast. So it's not looking good. If you look at the last two things that I was talking about, which is tauopathy and proteopathy, tauopathy means that you can't get from A to B. Nutrients can't, it's not going to have all the stuff that the cell needs to respire or work, and you have a tangling of microtubules which kills the neuron. And in proteopathy, you have the proteolysis of APP, and that sees the misfolded enzymes produce insoluble neurotoxic beta amyloid deposits over here, which basically kill the neuron completely or kill other brain cells. So let's go back to what we saw at the start. What happens in someone with dementia? I already told you that to remember something, you travel down this network of neurons, and you have specific amounts of neurotransmitter that are transmitted and therefore you can like trace your steps back in the sand along the beach or whatever. But in someone with dementia, these neurons don't look like that, they look like that. And because they look like that, these neurons are going to misfire. They're not going to be well insulated, they might not even report the amount of neurotransmitter that they have or are accurately working or firing, or they might even not work at all. And because of that, you're not going to have this flawless memory trace from A to B. It's going to be more difficult for you to find your memory or locate it in your brain. Again, going back to the beach analogy, you can imagine this as walking down a beach, and then waves come along and wash away your footsteps, or someone comes along and walks along your footsteps. And you're looking back at your footsteps and you want to retrace your memory. You want to see, where are those numbers. But the thing is, you can't do that because the memory's still there, but the neurons and the way of you getting to it is completely muddled up. You and your memory are completely unlinked. You won't be able to find out what that memory is. So here's a picture of the brain, and this is what happens with people and the progression of dementia as it gets worse. Normally dementia goes from the outside in. So it goes from the outside and then goes inside the brain. And you see a global loss of, firstly, frontal lobe, like the frontal lobe sees a loss in activity and all that, and then it goes inside. You've got the higher mental functions that are disrupted first. So you might not be able to concentrate very well, or plan certain things, or make judgments, or express yourself emotionally. And you might become more disinhibited. You might become slightly more racist, or something like that. And then over here, then it spreads across the whole of the outside of the brain, and you might not be able to control your eyes properly. You may not be able to hold things as accurately as you were once able to. For instance, you might not be able to hold a glass of water. Also, you may not be able to sense things properly. So the fact that I'm holding this right now, I probably wouldn't be able to do that. I might not be able to regulate where I'm pressing buttons. Or you may not be able to associate the senses properly, or recognize people, recognize pictures, and that's classic symptoms of dementia. And as time progresses, all of those things are going to get worse because neurons are being eaten away from the outside in. And then later on, this is a bit more advanced. It starts hitting this area here, which is the emotional area. This governs the fight or flight response. And this is where people can also start getting quite vulnerable, because if they don't have that fight or flight response, or if they can't notice that there's an attacker coming at them from down the street, trying to rob them, what if they try and give stuff to that person, or they don't notice that that person is a threat to them? They can be vulnerable and open to abuse. And the very late stages of Alzheimer's means that it might actually start hitting the cerebellum, which is quite far into the brain over here. And this means that they might not be able to walk, or their posture will be wrong, they may not be able to coordinate movement, they may not be able to move their muscles properly. Stuff's going to go pretty bad from then on, and then afterwards, if it hits the brainstem, that's the last part, because that's basically paralysis, death, end of. The University of California did a study. They took MRI scans of a range of brains, 6 months apart for a period of 18 months. And they found out that there were up to 40% less brain volume in these blue areas over here, and that the hippocampus had actually increased in brain volume, maybe up to about 40%. And as we can see here, that's a big problem, because this doesn't mean that the hippocampus has become larger. It means that there's a lot more deposits of beta amyloid fragments in the hippocampus. And since the hippocampus is the main thing that it's the main area of the brain that consolidates short-term memories into long-term memories, this explains why people with dementia can't figure out or make new memories about what they did 2 years ago or something like that. Furthermore, you have minus 10% of total brain volume over the 18 months. And this is universal. You can see here that the whole of the outside brain has basically completely decreased. This means that 10% of the brain, that's 10% of your memories, that's 10% of the way you work. That's really, really bad. You have two unrelated scans on top of it here. The first scan shows somebody with really early Alzheimer's, or somebody who's maybe healthy even. And on the left you can see, maybe not very well because of the board, but you can see that the outside of the brain is completely gone. It's completely gone. And that the only bits left are these bits over here, which are the inner bits of the brain, the inner parts of the brain, responsible not for the higher mental functions. So this person is basically going to be a diminished person, and it could lead to them not having basic functions or even die. So I'm sure here's the bit that you all came to see. How do we cure Alzheimer's? Well, there have been studies going in hamsters where they've transfected tau proteins out from other organisms and then put them inside the hamster, and they've managed to locate a specific kinase, PRK, on the endoplasmic reticulum, which regulates phosphorylation of the tau protein. And the thing is, during mitosis, you're going to have the cell noticing that something's not right, and it's going to kill the cell and undo the tau proteins. But if you inhibit this protein, that means everything becomes a bit better. You're not going to have the kinases destroying the tau proteins, but you're still going to have these other routes over here of reaching cell death. Another way to do it is that there's a branded drug right now called Taxol, or NAP10, which the Alzheimer's Society in the UK tried to use on fruit flies. And this is really good because earlier I was saying how the tau protein dissociates itself from the microtubules, and the microtubules just break away. But using Taxol, it actually binds the microtubules, a bit like sellotape, when the tau hyperphosphorylates. So even when the tau dissociates itself from the microtubule will still be there, it'll be fine, it'll be working, the nutrients will still go from A to B. And the next thing is that they're also looking into inhibitors for beta and gamma secretase, so that you don't actually get beta amyloid in the first place. But there's a problem with all of these at the moment, and it's that you're inhibiting something that's natural to that person's body. You're inhibiting these enzymes, beta and gamma secretase. You're stopping microtubules from dying when they should. And that might have unintended consequences. Because now, if you inhibit beta and gamma secretase in somebody with dementia, you're not going to have any secretase enzyme at all, and that might have unforeseen consequences that we don't know of. So, in the future, how can we actually cure dementia as a whole? Now, this is quite recent. About a month or two ago, a new drug called Glybera, which is an gene tipavirat, was recently approved in the EU, and this is the first time gene therapy is available on the global market. And this works by synthesizing the gene and splicing it inside an adenovirus carrier. And then this virus basically infiltrates its way into the nucleus of somebody with Alzheimer's, and then the cell starts making proteins using that gene, biology. So you get no more proteins, you get no more plaques, and you get no more tangles, because the cell's now making enough secretase. The cell is now making proper ABP, proper tau proteins. And it actually alters the DNA completely of Alzheimer's patients. So this has the potential to completely eliminate Huntington's, which is a genetic disorder. But obviously this is still very much in the future, because we have to get over the ethical issues of sticking foreign DNA in somebody on purpose, basically. And let's get back to the numbers from the start. Put your hands up if you can still remember those numbers, maybe. All of them? Right. So let me see. Okay. I'll pick someone then. Sam, do you still remember the numbers?
Yeah,
Yeah, go on then.
15, 16, 42, 58.
Wow, well done. That was excellent. You've got really, really good short-term memory. Or it might possibly be possible that you've seen Lost before and you remember the numbers 4, 8, 15, 16, 23 and 42, because those are like everywhere in that. And you only needed to remember the numbers 1, 3 and 58. But that shows that you're using your short-term and your long-term memory, because your long-term is what, maybe a couple of years ago when you were watching Lost for the first time, you had, assuming you have actually watched Lost, right? You committed those numbers into your long-term memory through your hippocampus. But somebody with dementia can't do that. So, what if you had dementia? How long would it take you to remember those numbers? Well, in the average person with dementia, so I know this is kind of weird because you can't really say there's an average, but maybe it would take 5 to 10 minutes, right? But it would take them longer to remember the memories in the first place because the neurons misfire, the hippocampus is completely destroyed, and the memory is wrong. But how long can a dementia patient's short-term memory even become? Any takers? How much time do you reckon it would take? A minute? Somebody? A minute? Well, actually, in patients that are really advanced, they might not even have any memory whatsoever. You may tell them something, but what's this talk about? And they won't even be able to remember. They might have to ask that question again and again and again. That'll be the big problem. So, right, I hope you found that talk really interesting and useful. Thanks for listening. No? Okay.
Yeah, what do you think are the implications of dementia on society
Well,
in general?
dementia as a whole at the moment, we don't have any cures for it. So people get stuck in care homes, they get put away from their family, and it costs like the NHS quite a lot of money to deal with. At the moment, you've got people who don't really know how to deal with other patients who have dementia. I mean, that you're going to tell somebody, oh, it's easy to remember something, but the person who has dementia obviously might find it difficult. It might be a bit difficult for them to remember things and all of that. It's got massive consequences. And like I said, in 50 years' time, 2050, you have, what, 8% of people are going to be over the age of 65 or something like that. You're going to have dementia everywhere, so we've really got to accelerate this gene therapy.
I was going to say 1% of global GDP is spent on treating people with neurodegenerative diseases. But my question is, what do you think the future for gene therapy will be? Do you think it will be a good?
Well, in my opinion,
treat many of the diseases we have today, or do you think it will something else will come along?
well, in my opinion, I think gene therapy is going to be really good for Alzheimer's and dementia because you bypass all these other problems that you have with inhibition of enzymes and all of that. But it's a slippery slope, isn't it? That's the main argument against gene therapy, is that once you start curing dementia, then how long until you maybe have a gene that gives you bigger muscle mass or something like that, or I don't know, if you're bored, it gives you more hair, you know, there's differences. So,
Chris, on the point of GDP, obviously a lot of money goes to the treatment of dementia. I don't know if you talked about this already. The presentation is looking at it from a slightly different perspective. But to what extent do you think there's sort of manipulation by big pharmaceutical companies to delay the progress of dementia care? Because obviously these kind of gene therapy, that kind of stuff, it's been around for a very long time, it's never been implemented. Do you not think that, in an extent, especially in the Americas, there's massive manipulation by pharmaceutical companies to delay dementia treatment in order to make more money by, treating the symptoms rather than the disease itself?
well, yeah, I definitely think so. That's kind of how medicine works today as well. When you have somebody, you send them home after they just get better the first time. You don't wait until they're totally cured. And gene therapy is that cure-all and end-all. Once you give somebody a new gene, that's going to stick with them. They're cured. They're not going to need to go to a care home. They're not going to have that 1% GDP globally spent on care in the community and all of that. It's a do-all and end-all. So yeah, I think big pharma, they might be doing stuff to manipulate, delay gene therapy, and on top of that, you've got that big ethical question: how far until we draw that line? When do we stop saying that gene therapy is good and big pharma are using that to their advantage? I think. Great. Thanks.
Guys, just a second.